run. So today I'm to give you a very short presentation on our research into treatments for muscular dystrophy and other inherited diseases. Now, Now, what I thought I would put up first, Raphael mentioned an illness called severe combined immunodeficiency. And this young man here, he's now 11, his name is Rhys Evans. And Rhys was born with this inherited immunodeficiency, which a clinician would tell you now it's an illness which is basically incompatible with life. And Rhys was treated by a gene therapy, indeed a combined genetic and stem cell therapy, at the age of a few months, single therapy, and here he is now on nearly his 11th birthday with his younger brother and his mum and dad, leading effectively a normal life. This is the power of the new genetic and stem cell therapies that are coming through in the pipeline. And the sort of illnesses that are being targeted go from the relatively common neurodegenerative diseases, diseases affecting the nervous system, to the cancers, to many of the rare inherited disorders, to infection and immunity, and to the almost epidemic levels of cardiovascular disease that we have in our modern day society. Today I'm interested in talking about Duchenne muscular dystrophy. You've seen this picture already. Raphael showed it of a young man with this illness. Now, our understanding of these diseases has been developed through a huge scientific endeavor called the Human Genome Project. And the knowledge of the sequence of the human genome has led to our understanding of the illnesses and enabling our ability to search for treatments. Now, Duchenne muscular dystrophy is an X-linked muscle disease. That means the gene that's affected in Duchenne muscular dystrophy is on the so-called X chromosome. And all the ladies in the audience, you all have 2 X chromosomes. And all the gentlemen in the audience, I'm afraid you only have 1 X chromosome. And if that X chromosome has a damaged gene in the context of Duchenne, then the boys will get the illness. Whereas for ladies, the damaged gene is compensated by the other normal copy, and they are, in a sense, what are called carriers of the illness. They can pass it on to their sons. So Duchenne is an X-linked disease affecting mainly boys. Now, as I said, it's a disease, a degenerative disease of muscle, the skeletal musculature. This is an image of basically 4 muscle fibers, human muscle cells, and the students in the audience will recognize this striated pattern, which is the molecular contractile apparatus that allows the cells to move and allows us to move. And the protein that's encoded by the Duchenne muscular dystrophy gene is called dystrophin. And if we turn these fibers round so they're coming at you out of the screen, and we look for where the dystrophin protein is, we find it's located here, actually coating the inside of these muscle fibers. And in Duchenne muscular dystrophy, this protein is missing. The gene is faulty. And in the absence of the dystrophin protein, the fibers become smaller, they become fewer in number, and the muscle undergoes a sort of fibrotic or scarring pathology that you can see on the right-hand side. And that pathology is emphasized here also. So the fibers become leaky to materials diffusing in from the extracellular media. They have an inflammatory reaction. All these little dots here are immune cells in the muscle. And here we see the fibrotic tissue, the scar tissue that arises. And this illness manifests itself at about the age of 3 or 4. Up to that stage, the youngsters are relatively healthy and normal. And then mum and dad notice a weakening. They're not quite as active as their peers. And there's a very famous diagnostic tool. It's called the Gowers sign, which involves the boys going through a maneuver because they lack body wall strength to raise themselves from the ground. And here's another prospect. for audiovisual problems, but I have here a small movie just showing you a youngster demonstrating or going through this Gowers maneuver to get up from the floor. So this is one of the first signs of weakness in the boys with muscular dystrophy. And as the disease progresses, it's a muscle wasting and weakness illness. The boys show this weakness. They then need physical support to their legs and the body trunk. Eventually, in the early teens, they're what's termed wheelchair bound. They need a wheelchair to move around. And then in the later stages of the illness, the breathing muscles are affected, and this becomes an extremely life-threatening and debilitating condition. So what can we do at the moment for boys with Duchenne muscular dystrophy? Well, there are very few, in fact, really, there are no effective conventional drugs. So this is effectively an untreatable condition. Steroids are given, but the side effects are extremely severe. So I'm going to describe 3 approaches to therapy, if I have time, very quickly. Firstly, what's called exon skipping. Forgive the terminology, it's an attempt to correct the genetic machine. And then gene addition, which is an attempt to add a functioning laboratory-based healthy gene for the patient. And finally, genome surgery, which Raphael briefly mentioned. This is a new technology to directly go in and correct the very Duchenne muscular dystrophy mutation. Now, exon skipping is based on the fact, some of you will know this if you're in your A-level studies, that certainly in eukaryotes and mammals, genes are not a single continuous unit. They're made up of fragments that are brought together. Those fragments are called exons. Imagine them like a linear jigsaw puzzle. You have to join together all the pieces to get a functional gene. And the idea with exon skipping is to step over, to mask damaged or unhealthy exons, to step over them, as shown in this diagram, and thus, in a sense, correct that damage. Now, in muscular dystrophy, there are actually 79 pieces of that linear jigsaw puzzle. They're all shown here, 1 to 79. And this is a healthy individual. In the case of Duchenne muscular dystrophy, for example, the jigsaw pieces, shall we say, from 45 through to 50 can be lost, can be deleted. And when the genetic machine tries to join 44 to 51, you can see they don't really match. However, if we can mask that piece, that 51, then you can see that 44 and 52 actually do match together. And this gives a sort of corrected gene, which gives a milder subclinical phenotype. That's what we're trying to do with this antisense therapy. And it's running in the clinic at the moment, here in the UK and elsewhere. And there's a consortium of scientists and clinicians working on this. And to give you a taste of what's been done here, firstly, we did a study where we simply injected the antisense drug into a small muscle for safety studies. And then secondly, there was a study where the drug was infused through an intravenous line into the patients. So it went to, in a sense, all the muscles in the body. And you can see that muscle is a big proportion, a very important tissue in the human body. And the outcomes from that, very briefly, were very positive. In other words, this protein, this dystrophin protein that I've shown you earlier, which is absent in the patients, was found to be restored. Here's a very faint example, but here's 1, 2, 3, 4 other positive responses in the patient, and here's a slightly less magnified image. So you can see a very positive response here, where we had up to about half of the dystrophin positive fibers restored, and about 20% of the normal protein levels in those tissues that were examined. Now, I show this with a little bit of trepidation. Where are we leading with this study? Well, here I'm going to show you the outcome of a veterinary clinical trial. It was conducted in Tokyo by these gentlemen that are shown here. And what I'm going to show you, two movies. The top movie is a dog which has been diagnosed with identical disease to Duchenne muscular dystrophy. Now this is an untreated animal, and you can see that he's a happy dog. He's not in any pain. He's a little bit slow. He gets a little bit weary. He needs a lot of encouragement to walk up and down the corridor here in this veterinary facility. And eventually he makes his way back down the corridor with a lot of encouragement. You can see that here. But he's slow and he's tiring. And his muscles are weakened by the very same identical genetic disease that we see in the boys. So there he is. And below we have a video which follows the same dog or a similar animal diagnosed with this form of muscular dystrophy who's been treated with a number of injections of this antisense drug that we are now trialling in the human clinical trials in boys with muscular dystrophy. And you can see that this fella is fitter. He's not perfect. He's fitter, he's stronger, he's happier to run and move around. And an outcome of this type in human clinical trials would be fantastic achievement if that proves to be the case. So we're in exciting times with this technology of individualized medicine, new landscapes of regulatory and safety testing, because some of these medicines are almost individualized to specific patients. And those trials are ongoing, and one or two pharmaceutical or biotech companies are involved now, and we hope that we'll have some sort of validation of whether this really works or doesn't work in a year or two's time. Now, with this type of therapy, only about half or maybe 60% of patients with muscular dystrophy are eligible for this type of therapy. So we need other potential treatments, and that takes me very quickly to this gene addition therapy that I mentioned. Now, what this is based on is the so-called recombinant DNA revolution. You'll have, some of the students, if they're biologists, will have heard of this, where genetic material can be isolated and grown in bacteria in large quantities. And really, this was where I, in a sense, came into this field, was to produce the first laboratory-based muscular dystrophy gene. And we simply did some injections into patients, and the outcome is shown here of effective but low efficiency correction of the muscular dystrophy deficiency. Now, I was a lot younger when I began some of these studies, not quite as young as is shown in this slide, but I just wanted again to show to the students that from the point of being at university, and if you can't believe it, that is actually me, at university in Glasgow, where life was pretty tough, you can tell from the background. Evolving into a creature like this some 40 years later. And you have to think for yourselves, if you're going to university, where will you be? Where will this science be in 40 years' time? I predict there will be huge advances in all of these areas. So the sort of advances we've made is to improve the delivery of these new genetic elements, these new gene therapies. And to do that, we use virus, vectors. These are disabled, safe, non-pathogenic viruses like the one that's shown here up on the left of the screen. These are actually beautiful organisms, actually. For any mathematicians in the audience, they're structured as dodecahedrons, 20 triangular facets to the virus with various apexes. I hope you can see that. But using this type of system, we can deliver genes at high efficiency to skeletal muscle. That's shown on the right. We can show by various techniques improvement in the disease, reduced inflammation, reduced leakiness, improved muscle function. And this type of technology is now being scaled up to pharmaceutical level to be taken through into, again, clinical safety and testing trials. So there's a perspective for this so-called classic gene therapy strategy. We have the genes, we have the delivery system. This could be a one-off treatment like I described for Reese earlier on, or at least long intervals, and it would be effective for all DMD patients. So we need a few more years to try and get this at least into clinical trial. So gene addition therapy. Finally, just very briefly on this. I'm sorry, I believe we're very short of time. So-called genome surgery. This technology, as Raphael described, takes advantage of new genetic engineering techniques where we can produce enzymes which will actually recognize very specific genetic sequences, i.e. the Duchenne gene. They will cleave the chromosome, open it up, and they will then replace any damaged regions by a replacement genetic segment. Now, in the case of muscular dystrophy, there's a prototype shown here. You can see at the top, the jigsaw parts 45 to 52 are missing. We could use genome surgery to open the chromosome, and we would open the chromosome right where the DMD gene exists, and we will then introduce the missing pieces of the jigsaw as a single unit to give a corrected full-length product. So this genome surgery perspective is really a new landscape. It's actually a sort of revolution in biology. But the systems exist and the repair matrices exist, and this will be tested in stem cells in skeletal muscle. And just very briefly, for those of you who haven't really seen a stem cell, these are muscle stem cells, stained and histologically in various ways. And again, if this will run, I just wanted to show you this. This is another one of these muscle fibers. And the sort of little bumps up and down in the background in this fiber are the stem cells. And those stem cells are normally quiescent, but if they detect damage in the muscle fiber, well, it's not going to run, I apologize, that's the switch over. But they will run, and I could have shown you them moving up and scurrying up and down the fiber, looking for damaged areas in that system. So we're in a phase of translational medicine here, from original discovery in 1953 through to a medicinal product. This work requires partnerships in finance, patient groups like the ones that are here today, universities and hospitals, all the way through to the biotechnology companies that will put the medicine on the pharmacy shelf for a doctor to actually prescribe eventually to patients. And there's lots of acknowledgements here. Some of these people you'll recognize. They've been showing you around the labs. I think two of them at the front here. Just a thank you to the people in the lab, my lab, who do this work. There's some of them all sitting in the audience here, smiling at themselves. Thanks to the patient groups, the funding agencies that make the work possible. Action Duchenne are here today, and Diana's going to speak in a moment. Muscular Dystrophy Campaign are here today, and many other agencies that contribute to our work. And just finally to say, it's a lot of fun doing science. This is the summer picnic, the lab picnic in Windsor Great Park, not far from here, so that we don't always wear lab coats in the Coughley series. It's fun doing science as well, and I would encourage you to think about a career in that direction. So I'm finished there. Thank you very much.